So hopefully you were able to figure out the third difference of RNA from DNA by looking at this strand. But let's recap the other two differences real quick. Number one, RNA on the side strands, instead of having a deoxyribose sugar like the DNA has, it just has a ribose sugar, also attached with a phosphate, same thing. And then the second difference is that RNA will only be found as a single strand. You will never find a double helix, double-stranded RNA. Never, ever, ever. DNA obviously is double-stranded, twisted into a double helix, except when it's unzipped by helicase, right, and it's being replicated like we talked about last week. But RNA will never have that going on. RNA's job, it has a couple different jobs, but its main job is to take this code from the DNA, as we're about to see, and take it out of the nucleus, because that code cannot be read in the nucleus. So this is exactly what it did. Now that third difference is that there's no T. RNA does not have thymine. It has uracil, and we represent it with U. But on the RNA strand, it acts just like the T acts on the DNA strand, meaning it always matches up with A. It never matches with G or C. It always matches up with A. So it's just a little bit of a difference in the molecule that is being attached. So all you really have to remember when you're thinking about RNA and you're matching the pairs is that U will always go to A. That's what you have to remember. Now, when this is copying this DNA strand, like it did over here, it has a very specific name. It's called transcription. And that's a very important name because there's two steps in this code reading. That first step is transcription. It's for that RNA molecule to get in there and read one side of that gene. Now, it's not reading the entire DNA strand. Okay, this isn't replication, where helicase is coming in and unzipping the entire strand and DNA polymerase is coming in behind it and matching both of those entire links of the strand. That's not what RNA does. RNA comes in and all it's worried about is a specific gene because the cell is saying, hey, I need a protein and I need a specific one and I need it now. So messenger RNA runs into the nucleus, goes to that gene, which remember is a piece of the DNA segment. So the strand of DNA can be millions upon millions of base pairs long. Maybe that gene is only 20 or 30,000 base pairs long. Maybe it could be 100,000 base pairs long. It depends upon what it is. But the RNA will recognize that, will go in and grab it and just copy that piece of the gene that it needs the protein for. Again, this is called transcription. Transcription basically just means to copy. Now, after transcription is finished and we have our copy of the DNA strand, now our RNA is going to leave the nucleus. It's going to go out. This strand is called messenger RNA. We abbreviate it mRNA. And that's exactly what it is. It's a messenger. If I needed to tell someone something down the street, and I couldn't go down there myself and my cell phone was broke, after I quit crying because my cell phone was broke, I would probably send a messenger down there. Okay, maybe I would tell my boyfriend, hey, can you run down and tell so-and-so what I need to tell them? That's exactly what messenger RNA is doing. It's taking this code, it's copying it, and then it's going to take it to the ribosome so the ribosome can then read this code, and we're going to talk about that next. So here's my messenger RNA, mRNA, and out the nucleus it goes.